Hi, good evening. My name is Josh Newby, and I'm the founder and executive director of Teresa's Research Foundation. And I also work at the Lester and Sue Smith Breast Center and Baylor College of Medicine. And today I'm honored here to have Dr. Alana Welm from the Huntsman Cancer Institute, who's an endowed chair in cancer research. It's a great pleasure to be invited here to speak with Alamo Breast Cancer Foundation as they're kicking off a San Antonio Breast Cancer Symposium. And they've asked us today to have a chat about metastatic breast cancer research. And so couldn't think of a better person to bring along with me than Dr. Welm. And so what we're going to do here is just kind of have a more of a casual conversation. As you know, you're always so inundated with slides and so many scientific figures. I thought it better to just have a chat today. So I'm kind of going to send a lot of rapid fire questions your way, Dr. Welm. So just if you need me to repeat anything or go back or slow down, let me know. But I'll try to not put like 5 questions in one as much as possible.
Okay, it's my favorite topic. So
Yeah. So I think it's better to start, as everyone's in a different place in their knowledge and understanding of metastatic breast cancer and metastasis in general. I think it's just a quick kind of, 30-foot view of kind of what metastasis is and why it is so important we study it.
yeah, sure. So metastasis is when the tumor spreads from the breast to other parts of the body. And this is what causes death from breast cancer, unfortunately, because once these cancer cells spread to the liver or lung or brain or other places, they become really hard to treat. And I think that that's one of the puzzles of metastatic breast cancer is, why are these tumors so much more drug resistant than the primary tumor? What is it about their microenvironment that's allowing them to survive? How do they hide out in the body despite treatment for sometimes decades before they pop up? And I think that though it is important to point out that metastatic breast cancer, even though it's not curable, it is treatable. And we have people who are living longer than ever now with new treatments for metastatic disease, but we still have a lot of work to do so that we can make this curable.
Right. And I think that's a good point that you bring up. I remember, a few years ago at San Antonio, you presenting and you had that slide. And of course, I always bug you about it, but I think it really brought home that you mentioned this progress we're having, but the statistics are showing that, more people are dying every year of metastasis and especially of metastatic breast cancer. And the impact that's having on families. And of course, now we're seeing young families and issues of, of course, disparity as well being impacted by that. So what do you have any kind of insights on why we're seeing that? And I think maybe why that's such a concern.
Well, I think people are living longer and longer with metastatic disease. So that's a good thing. But because it's not curable still, then those people usually will eventually succumb to their breast cancer. I think, we're getting better at detecting whether it's the initial tumor or the metastatic tumor. So that probably has some role in the, amount of cancers that are diagnosed and treated and the number of people who end up dying of the disease. But ultimately the problem is we just don't have therapies that can, eliminate the tumors once they're metastatic. And I think that's the holy grail. That's where we have to go.
Right. And I've heard this mentioned before by yourself and others, essentially we run out of drugs. We get to a point, it doesn't matter where we're at in the science or with just run out of drugs. And so I think that's again goes back to why this area of research is so important.
Mhm. Yeah, that's right. And I think, one of the interesting phenomena in breast cancer, and I'm not a physician, I know this from a science perspective, is that as people live longer and longer with good treatments for their, metastatic disease, we're seeing more and more brain metastases, which are harder to treat because many of those drugs don't cross the blood-brain barrier, et cetera. And so now we're seeing sort of a shift in more and more brain metastasis relative to other types of cancer. And we have had some nice advances, especially in HER2-positive brain metastasis that was reported a couple of years at San Antonio. So I think there are advances. It's just that, we have to keep up with these tumors. And I think that there's really 2 areas where we can do that. One is we can find better therapies that can overcome this drug resistance that we see in the metastatic setting. But we also can think about ways to prevent metastasis, and that is ways to kill these cancer cells that have spread in the body, have not yet started to grow or be detectable, but are sort of in this minimal residual disease state. Where if we could find therapies to either kill those cells or keep them from growing, we would prevent metastasis essentially, and that could have a really big impact as well.
Right. So that's a really good, I think overview. And of course, we can provide links for people to kind of dive more into the research. But I think now I kind of want to get a bit into the weeds a bit. This is an area of mine that I'm really passionate about is really looking at model systems in research. So there's so many different types of models and areas that we could kind of go into. But I thought maybe since I'm lucky enough to have you here, that we could kind of talk about two areas that you really specialize in, which would be PDX models and then organoids as well. And so maybe if you could kind of just give a brief background of each and maybe kind of give that highlight.
Sure. Maybe I'll start with just a little bit of a historical perspective. So, for many decades, we've been studying breast cancer, human breast cancer, by studying cell lines that were developed many decades ago and have been grown in the lab ever since. And the technology has just evolved so much that now we can actually study contemporary patient tumors by collecting tumors from patients and growing them in the lab in different ways. So probably the earliest way was to grow them in mice. So we would implant these tumors into mice that are genetically engineered to lack an immune system so that they can grow a human tumor in their body. And these are called xenografts, so patient-derived xenografts or PDX models. And then things have also evolved now to have a much more cost-effective way to grow these tumors, where instead of growing them in a living animal, we can grow them in organoid cultures, which are these three-dimensional gel-like cultures that we can put the tumors in. And believe it or not, they actually organize really nicely in those settings and show a lot of the same properties that they show whether they're in the mouse or in the human, including things like response to different drugs, et cetera. So I think that what this allows us to do is study contemporary breast cancers. In other words, tumors that are isolated from patients that have undergone today's therapies and have developed resistance to those therapies, and try to find new therapies or try to understand why they're resistant. There are caveats, of course, to any model system. And one of the big ones is that by studying these human tumors, whether in an immune deficient mouse or in 3D organoid cultures or in other ways in the lab, we're really missing a large component of the environment that the tumors would normally be in while they're in the human liver or human brain or other places, for example. So one of the challenges we have, for example, is we don't have a functioning human immune system that we know is important in terms of regulating the tumor progression and studying things like immunotherapy. So we still have more to do in modeling human breast cancer, but I think that the technology has come quite far.
Yeah, I mean, it's really exciting to kind of see, just even in my short time in doing all this, where or organoids have come on, because I think you highlighted so much about with PDXs. I mean, you don't, and others don't like to talk about it much, but it's a really hard technique that you've spent years trying to perfect, and are always clear to say that it doesn't always work. But I mean, I think it's exciting to see the technology moving forward.
Yeah, exactly. I think one of the challenges, of course, is the cost of these types of models, and they grow slowly, just like tumors grow slowly in people. And so, it's not the favorite type of project for a graduate student. Let's just say that.
Right. So I think another, area that I'd like to kind of give a little bit of background on is a bit about what you're doing in your lab. I think because metastasis is such a large area, and maybe just kind of narrowing in a bit, because I think it could give people an idea of kind of the types of research that you're able to do in the lab today.
Yeah, we really have two major areas of research in the lab. And maybe first I'll focus on the patient-derived models part, since we started talking about that already. But if you're interested, I can also talk about our tumor immunology
Oh yes, we're
side of
definitely
the lab
we
as
have
well.
to talk about immunology. I think with especially with San Antonio coming up and just coming off our conference, so we got to
Yeah,
get both of
okay.
them in there for
So
sure.
I'll be brief then. Yeah, that's also very exciting. So in our patient-derived models, cells, I think the most interesting and exciting thing we found so far is that we found that breast tumors from patients that can grow in these mice. So we're talking about newly diagnosed breast cancer patients. We take the tumor from the breast and we put it in a mouse. Only about 28% of them will grow. And the most exciting thing we found completely accidentally was that those tumors were the ones that are the most aggressive, and they will recur as metastatic disease in the patient, and the outcomes of those patients are really bad. So what we started to do then is say, okay, if we have this person's tumor growing and we know that they're at high risk of recurrence and death from breast cancer, we need to do something about it. And so that's why we developed the organoids, actually, was so that we could do some medium to high throughput drug testing on the individual patient's tumor material in order to personalize therapy. So we have a couple of trials that we've started here at Huntsman Cancer Institute, and we have one open now for specifically for metastatic breast cancer, where we try to make organoids from the patient's tumors and test drugs in order to personalize therapy. So we're really excited about that. It's a feasibility study. We still don't know yet how well that'll work or whether it will change any outcomes, but we have to start somewhere. So that's one part of the lab. Do you have questions about that before I talk about the immunology?
Yeah, I mean a bit, because I think that was something that this project that excites me so much. And I remember, a few years back when you started mentioning it, and it's been so many years I know that you've put into this and the team. But a lot of times, the feedback that I'll get from someone is like, oh, basic science, it takes so long and I'm never going to see an impact from it. But I think this is, though it's still really, in its early stages, this is something where you're kind of seeing something happen really in real time, which is really exciting from the research perspective.
I think it is exciting. I mean, to be fair, it has taken a long time. We made our first PDX model almost 15 years ago, so it's been slow and taking time. But now we are actually able to return precision medicine results back to the patient, and we'll see what happens. I think it's really exciting. One of the challenges we have, though, are things like regulatory issues, right? So normally, if there's a test for anything that would direct patient care, that needs to be done in a very specialized laboratory setting, and we're just a research laboratory, so we're not yet certified to do those things. It's a little bit of a catch-22, right? Because you have to first show that it's feasible and it's effective before you can go through the effort of certifying your test, and yet in order to do the trials, you sort of need to certify your tests. So we've been kind of trying to sort of cross these hurdles as we come to them, and it's not something we've done before, or I think most people have done before. And so it's been a little bit challenging, but we were able to find a good medium where with our IRB that would allow us to return results to the clinic, as long as we disclose that we're not a CLIA certified laboratory, that these are research results for a research study, and we were able to move forward with it that way.
Yeah, I think it's really important to highlight, all these different regulatory issues and things that exist. And I think especially with technology moving at such the rate it is, there's not these things in place to kind of handle this, and so, I really appreciate, all the work that you and your team have done to kind of, try to blaze this trail a bit. And, I know, for example, at Baylor, you recently presented some of this work to all of our group, and everyone's really excited, and there's others as well. And so, I think it's really, really, brings some hope that we can really gain some knowledge from this work. So now I think, to really get people in, we got to talk a little bit about some of your immunology work, because I think that's really exciting. And of course, everyone, I think kind of has an understanding a bit about the immune system, and that it's definitely playing a role in cancer.
Yeah, so our work in immunology has really evolved again over many years. We study this particular protein called RON kinase, and we found many years ago that it was causing metastasis in mouse models of breast cancer. And as we chased that down over the years, we realized that the reason why it was facilitating metastasis had a lot to do with its role in the immune system. And essentially what happens is this protein sort of suppresses the immune system in the mouse. And so now as metastases are trying to grow, the immune system doesn't come in and kill them. So what we found is if we knock out this kinase or use a drug to inhibit it, we can actually, even if the metastases are already seated in the lungs, for example, or in the bones, we can actually prevent them from growing. And the way that that works is they get completely swarmed by T cells. So T cells are part of the immune system that can kill tumor cells. And it's amazing when they do it because they just completely swarm these micro metastatic tumors and can eliminate them. So we've been working on, how does that work? What's the molecular mechanism by which this kinase regulates these T cells? And most importantly, can we, have a drug that can inhibit the kinase and, have some of the same effects? So all of this has been done in mouse models, and the reason for that is because we need a good, strong, functioning immune systems to study the tumor immune interactions. And as I mentioned earlier, our human patient-derived xenograft models are necessarily in immune deficient mouse hosts in order to allow the tumor from another species to grow. So we're also have made some strides in sort of putting in a matching human immune system with our PDX models, but that's still early days. We have our first paper coming out on that in breast cancer research, but it still needs a lot of work.
Yeah, I mean, it's just, I mean, so much work that your team's put into that, but I think it does, excite a lot of people because with metastasis and its ability to spread. I mean, getting the pleasure of hanging out with people like yourself, there's just no way that the immune system doesn't play a role. And so trying to find a way through this kinase is very exciting, and look forward to that paper coming out.
Thanks. I think it's noteworthy to point out that all of our approved immunotherapies, whether in breast cancer or other types of cancer, are all focused on one particular function of the immune system. And there are many other aspects of the immune system that get regulated during tumor development and progression. So, we just because, as we know, immunotherapy has not been a home run in breast cancer. And in fact, it's not been very effective except for a very small population of triple negative breast cancer patients. Investigating these other ways that the immune system, gets suppressed by the tumor or can be reinvigorated to kill these cells, I think is going to be really important because there's a lot of other ways to do it besides the ones that have already been approved.
Yeah, it's really encouraging that, though some of these other areas haven't worked in breast cancer, involving, the immune system, that people still are saying, okay, well, let's look at some other ways to maybe do that, because it clearly is playing an effect on metastasis. And I think we've painted a picture here of how complex this problem is, and why we need so many people like yourself working on this in order to try to move the field forward. And so one way that we do that, and I guess full disclosure of your and I's relationship is, we've been working together for, gosh, I don't know, 5 or 6 years. We initially met through the conference that the foundation started. It's an annual conference for metastatic breast cancer research. And that conference is a research conference, and it's very focused on collaboration and partnership, very different than San Antonio, which is a large international conference for breast cancer, and has some really great basic science, but is clinical. We've evolved our conference to be a bit more research. But you and I have met through that, and it's been a real pleasure to work with you on that. And I guess we also found out, me working at Baylor, you trained and did your Ph.D. at Baylor, and I think the reason I highlight that is I think this world is so small that we all live in, and it's all interconnected, whether it be through advocacy and research.
I agree. Yeah, no, it's been a lot of fun. Working with advocates has been such a powerful motivator for me, not just for the reasons you would think, which would be, that it brings forward the heartbreak of breast cancer and how many people we lose, but also to motivate the research. Advocates have incredible ideas because they've thought about this from a different perspective, and I think that that really has changed the way I think about science. Even my team is now, every single one of them is engaged with advocates in their projects. So I think it's really a partnership that might not be a conventional one that we think about in science, but I think it's a crucial one.
Yeah, I think that this problem is so complex, as we've been able to portray in this conversation, and no one person is going to be able to solve this problem alone, and no one group of people. It can't just be researchers or clinicians or FDA regulators. We really do need everyone at the table working together to try to solve these problems. And so I think that having these different opportunities, whether it be through Alamo or National Breast Cancer or through what you and I do together with the conference, definitely are really important in moving forward.
Yeah, I agree. I think it brings a certain urgency to the research where, for example, one of my trainees might be presenting their project in front of our local patient advocate committee. And, they may be thinking about the mechanisms and how does this RON kinase work? And, how exactly does it regulate T cells? And, an advocate might say, well, have you tried, is there a drug that you can use to inhibit that? And the trainee might be thinking, well, I'm not quite ready for a drug yet because I don't yet understand every single way that this works. But the patient would bring the perspective that says, I don't need to know exactly how it works. We don't even know exactly how aspirin works, right? But we know it works. And so, it gives a little bit of urgency and a little bit of that little push sometimes to make people brave enough just to try something before you completely understand it.
Yeah, so that kind of brings up two quick questions I have for you in regard to that. So you mentioned this Huntsman group you have, and I know we've talked in the past that, and I've had the pleasure of meeting some of the amazing people that are a part of that group. They've been really helpful in getting some of like your precision medicine programs off the ground and helpful. Can you just talk about that just in brief about how important that's been to the work that you and the team have done?
Yeah, I mean, I first got introduced to working with advocates many years ago now in the DoD breast cancer research program, where I was introduced to the National Breast Cancer Coalition, and that's the first time I saw the value of advocates in not only participating or in discussions about science, but actually reviewing grants and really having a say at what kind of research gets funded. And it was just such an amazing, eye-opening experience that over the years I've thought, we should have a group like that here, right? That can help us hone our research. And around that same time, it just so happened that I met Dr. Carla Lloyd, who's a retired professor emerita from Syracuse University. She's a professor of communications and was being treated for breast cancer and had come in and said, I really want to give back somehow. Is there some way that I can be involved? And so when I met her, I pitched this idea and she really liked it. So she became our first member of our committee. She's now still the chair. This has been 5 or 6 years ago. And then she and I worked together to develop, what are the goals of the committee, a whole plan, and recruited more advocates. And now we have two scientific liaisons who are trainees in our programs here, and they serve as liaisons between the advocates and the science. It's so much fun, and we meet every month, never fail, and it's just really a fantastic group.
Oh, that's really wonderful. And I think it just shows the different impact that different groups have. And again, going back to that connection, I mean, National Breast Cancer, the impact they had on you as an investigator. And of course, I have to brag on you a bit as you were an Era of Hope awardee, correct, in the past,
Mhm.
which is a great grant for young investigators and has seen a lot of people move forward with that. But Alamo, of course, is a great ally of National Breast Cancer and does a lot of work in Texas for them. And I'm a project lead graduate myself and seeing that role. And so I think it's just kind of seeing again how these things kind of tie in and connect and why you need these different groups. I think sometimes people will say, well, why are there all these different people doing all these different things? Why isn't there just one? And I think it just again highlights the complexity of the program.
Yeah, I think the other important point, and you know this well because we have written a manuscript on it that we're trying to publish.
Yes.
But it's really important that the scientists more scientists see the value in working with advocates. And one of the interesting things is when we invite scientists to come give a presentation to our advocates, many of them are terrified. Like, what, because you don't, it is it's a different kind of dynamic, right? You don't want to say the wrong thing. And, all of us start our scientific presentations by talking about the mortality statistics of breast cancer, but that's difficult when you're a room full of people with breast cancer. And so I think teaching scientists how to interact with advocates and making them realize how much they benefit and their research benefits, and how much I think you can speak to this more than I can, but how much sort of reward an advocate gets from participating in that process, I think is really fantastic.
Oh, absolutely. I mean, I think, being a part of, a lab meeting or a grant, I've been blessed to be a part of some grants that have been successful and, I mean, $1 million grants. And it's just such an amazing part to feel like, you play a role in that. And I've sat in meetings where patients actually living with NBC are able to speak up and talk about, brain mets, as you mentioned before. And I think, have that real impact. And you can see that. And, I think, it's important that people understand that patient advocates aren't just a person living with the disease. They have so many different skill sets and expertise in their lives that can really add value to this. One last kind of piece on the advocacy thing is we just wrapped up our conference together in September up in Park City. And you mentioned your trainees, and I know they had a real. We had a small group together, having to be safe with COVID, but I think it was kind of even better in a way because we were really able to make some connections. And I know the few patient advocates living with NBC we had there, they made a real impact on a few of your trainees, and I know they participated in our good friend Julia's program Grasp, and that's been sounds like it's been really impactful in the lab there.
Yeah, and we got great feedback on that conference that next year people want to do an advocate trainee session where they can you sort of give an elevator pitch of their research project, get feedback from advocates. So not only feedback on the research project, but in a training venue to have our trainees learn how to present their research to more of a lay audience. Although the NBC advocates are very educated about this disease.
Yeah, absolutely. I think it's exciting, and I'm looking forward to it. So, as our kind of time is starting to wrap up here, just a kind of quick, a little more rapid fire questions for you. So,
Oh.
um, you highlighted a bit of the lab's work and things like that. So, kind of what are your long-term goals or hopes, or even maybe just even a short-term goal as well for the lab, kind of the next 3 to 5 years.
Oh, well, short of putting myself out of business, which would be the best
Yeah.
outcome, cure breast cancer, or at least make it a chronic livable, disease. I think, our short-term goals are really to try to determine if this functional precision medicine idea has any ground. I think we really need to know, yes, we can grow people's tumors. Yes, we can test drugs. Yes, we can do all the genomic characterization. Does it make a difference? That's what we need to know right now, because we need to know how much to invest in this sort of individualized care. But we're really mindful of the fact that we're not going to be able to grow everyone's tumor. This is not, we want to develop things that will work for everyone, right? Not just
Right.
people who happen to live near a fancy cancer center or have money to pay a company to do this. So I think the longer term goal, if we can show that it's actually useful, is to develop biomarkers. So turn this very complicated test where we're growing people's tumors and personalized medicine into a simpler test that says, okay, your tumor has this feature, that feature, the other feature, and that means this drug will work for you, right? So that we extend it to where this could help people all over the world. That's my dream for the precision medicine part of it. We'll see.
Well, that's really exciting. I mean, I really applaud you for trying to tackle that, and like you said, trying to help everyone. I think that's what's really exciting, and really looking at that, because I think sometimes as an advocate, when I get approached by a newly diagnosed patient, and they'll ask me about it, and I really try to just, as I'm not a scientist, just kind of say, well, everyone's cancer is different. Everyone's experience is unique to their own. So many people will ask me, for example, about my mother, who succumbed to the disease. And then, like, that was a long time ago. So many drugs have been developed, and that was her story. Everyone has their own experience. And, the bottom line is, we have to kind of, keep working together and really trying to do this. So my last question is, so you've kind of given yourself some daunting tasks over in the future, and have given us a good background, but as it would be remiss to not, as we have San Antonio kind of coming up and kind of wrapping up the end of the year with our conference, what is other than your own work, of course, which I think has been really exciting with the publications that have come out and some of our friends and some of their work, but what's really got you excited about the end of this year, and kind of just where we're at and kind of going forward, that really can get people to get excited about?
Well, I'm really excited about immunotherapy still, but not the classic immunotherapy. As I was saying, doesn't need to be immune checkpoint blockade necessarily, but I think we're learning so much So that field has sort of opened up, and we're learning so much about all the different ways that the immune system is regulated, and it's different in every cancer. So I think the more we can learn about this in breast cancer, the more targets we'll have. And I really believe in the power of the immune system to take care of these tumors. We know it can work. We just have to figure out how. So I think that's what I think is going to be really exciting on the horizon. And then I think building these models of like I said, contemporary patient tumors, even if our precision medicine stuff completely flops and we just find it's not feasible, it's not going to work, or maybe it works, but we just don't have good drugs, right? We're limited with the number of drugs we can give to patients. So, but then we can use this big collection of models, which I hope will start to come from a more and more diverse set of patients through our collaborative networks, to discover new therapies. And as the technology is coming along, we're able to do more and more computationally. There's artificial intelligence integrating with the genomics and the drug responses. I think we're going to have an explosion of data that, if we can just harness it, should help us find all kinds of new targets.
Oh, well, that's really exciting. And yeah, I think it is so complex with all this technology moving so fast, and we're all trying to do our small part. It is kind of a real daunting task, but I'm glad that you're up for it. And I'm so grateful that to have this opportunity to chat with you today. A lot of times you and I are kind of more talking one-on-one, working on our conference together, stuff like that. So I'm glad that I kind of can share you a bit with others, and they kind of get to see some of the great work that you all are doing. And I hope people will be inspired to, maybe whether they check out the work that you all are doing, or just in general learn more about metastasis research, because I think it's a really important area.
Thanks. It should be a great conference. I'm looking forward to it.
Awesome. Thanks so much.
Thank you.